Hello, my name is Arpita Jaju, and the project I will be discussing with you today is the role of the genetic transfer of mobile genetic elements in the evolution of microbial populations. The lab in which I conducted my research, the Gogarten Laboratory at the University of Connecticut, uses computer coding and other bioinformatics techniques to investigate mobile genetic elements and draw evolutionary conclusions. My mentor, Shannon Susi, a graduate student in the lab, is working towards a comprehensive understanding of several different mobile genetic elements and their role in microbial evolution. I am working on the evolutionary relationships of one category of mobile genetic elements known as Group 2 introns, so my research will facilitate my mentor's research. My mentor provided me the framework of the experiment and access to several journal articles to gain a better understanding of its scope. She also provided me with tutorials for the computer coding and command line execution aspects of the project and encouraged me to use the wealth of information and tutorials available online should I encounter difficulties. Several of the coding scripts that were used in this project were ones that the lab had previously used, which I modified to fit the specific parameters of my project. Also interning in the Gogarten lab during the summer was high school senior Rachel Sun. She and I helped each other out when we came across problems, but we were each working on different mobile genetic elements. Traditional understanding of evolution has its roots in vertical gene transfer, also known as inheritance. It is the passage of genetic information from parent cell to offspring cell. However, Microbiology Review cites the recent emphasis in research on the horizontal transfer of genes, also known as lateral gene transfer. This passage of genes occurs between a donor and a recipient cell, which, interestingly, do not have to be related in any way for the transfer to occur. The sequences of genes being transferred are known as mobile genetic elements, and by tracing these segments of DNA through genomes of several prokaryotes, the Journal of Bioinformatics supports that one can find the relationship between them. As this study was conducted entirely through the use of computer coding, much of the 3-week process included debugging scripts and fixing errors. Additionally, NCBI's non-redundant database of genes was used to find similar genetic sequences using the Basic Local Alignment Search tool, or BLAST. Sequences were blasted for matches of 90% similarity because 97% similarity is the threshold for species delineation, and this would put results outside of the species cutoff, mitigating the effects of vertical gene transfer. However, results were often duplicates or were false positives. These had to be assessed and sorted out of the result pool. By the end of the project, I was able to cluster genes based on sequence similarity and construct a phylogenetic tree based on the horizontal transfer of mobile genetic elements.